Hello, everyone. My name is Trontaye Black. I'm a senior here at UNCG, and I work in Dr. Kern's Biodiversity Lab. My project studied the effects of nitrogen additions on mycorrhizal associations in Ambrosia psilostachya. Ambrosia psilostachya is commonly known as the Western ragweed, and the samples used in this experiment were collected from the Konza Prairie site in Kansas. The Tallgrass Prairie ecosystem is an important study site because it possesses high biodiversity, fertile soils, and is capable of undergoing carbon sequestration at high rates. Mycorrhizal associations are the symbiotic relationship between fungi and plants. These plants benefit from this because this symbiosis allows plants to absorb more nutrients in water, which encourages cell growth and leads to a healthier life. In addition to improving the overall quality of life for the plant, mycorrhizal fungi can also help their hosts adapt to changing climates, which has the potential to be a crucial buffer against climate change. Uncovering methods that would allow the Western ragweed to survive even under conditions of global change, such as increased nitrogen, is crucial to the symbiosis between the Western ragweed and Mycorrhizae and the overall ecosystem. From this background information, I hypothesized that nitrogen additions will have an effect on the rate at which the Western ragweed forms symbiosis with Arbuscular Mycorrhizal Fungi, also known as AMF. And this photograph right here is just a photograph of the Western ragweed. The soil samples and above ground biomass were collected in the summer of 2021. The above ground biomass was weighed and recorded while the root samples were washed clean of soil and then preserved in ethanol. The root samples were then cleared of cell contents to visualize mycorrhizae by submerging in KOH. Once the KOH solution was cleared, the excess KOH was drained into a waste container and the samples were rinsed with water. The roots were then stained with a recipe consisting of Trypan Blue, glycerol, lactic acid, and water, and added to test tubes. They were then soaked for 60 to 90 minutes at 90 degrees Celsius in the drying oven. The stained roots were then spread out on a petri dish and visualized and counted using the grid line count method under a dissecting microscope. This first picture is what the root samples look like prior to washing. The second picture is the above ground biomass being weighed. The third picture is the sample staining process, which occurred under the hood. And the last picture is what the stained roots look like on the grid line petri dish. The first figure, which consists of these three graphs right here, focused on the percentage of mycorrhizal colonization. The first figure on the top left is a box plot that displays the different percentages of the 4 nitrogen treatment groups. From left to right, the treatment groups are the control, which had 0 grams of nitrogen added, then 2.5 grams, 10 grams, and 20 grams. The figure on the right is a linear regression model of the mycorrhizal colonization percentage with the same 4 treatment groups from left to right, which will be the 0, 2.5, 10, and 20. The data table displays the p-values of this experimental group that was found using the ANOVA test, in addition to the 3 values of the treatment groups that received added nitrogen. The 2 graphs show that increased nitrogen had no significant increase or decrease in the percentage of mycorrhizal colonization among the different treatment groups, and the p-value of this group, which was 0.656, further emphasized that the increase in nitrogen had no significant effect on the percentage of mycorrhizal colonization on the roots. Now moving down to figure 2, it focuses on the above ground biomass. The first graph on the top left is another box plot that displays the average above ground biomass in grams of each of the 4 treatment groups. The graph next to it is another linear regression model of the same 4 treatment groups. The data table below, once again, displays the p-value of the experiment as well as the p-values for each of the 3 treatment groups. Looking at the 2 groups, you can see that there's a clear increase in above ground biomass in the treatment groups that received higher concentrations of nitrogen. The p-value found in the data table, which was 0.001, also supports that there was a significant increase in biomass as higher doses of nitrogen were added. Treatment group 10 and treatment group 20 each had p-values of 0.02 and less than 0.01 respectively, which reveals that these were the 2 treatment groups that had a significant increase in biomass. This tells us that an increase in nitrogen does lead to increased above ground biomass. And now moving over to the implications. It is important to understand the symbiosis between the Western ragweed and mycorrhizal fungi under conditions of global change because it plays a crucial role in the ecosystem. Understanding how climate changes affected the Western ragweed can allow us to generate a better idea of how it may affect other species as well. And this photograph right here is a photograph of the mycorrhizal colonization. And you can see that in the blue thin lines here. Yeah. And lastly, for my acknowledgements, I would like to thank my graduate student mentor, Sarah Gora, and Dr. Sally Kerner for their assistance, support, and guidance for this project. I'd also like to thank the UNCG Department of Biology for their undergraduate research grants, which helped cover the financial expenses of this project.